Good morning, students. In the last lecture, we were discussing about the reproduction in the bacteria. Reproduction in the bacteria, it takes place by the two methods. The first method, it is the asexual reproduction, and the second method, it is the sexual reproduction. Okay, in the last lecture, we have discussed about the different methods of the asexual reproduction. Asexual reproduction in the bacteria, it takes place by the binary fission, then by the formation of the cyst, by budding, then by conidia formation, and by the formation of the endospores. These are the different methods of the asexual reproduction in the bacteria, of which the binary fission, it is the most common method of asexual reproduction in the bacteria. Okay, in today's lecture we are going to discuss the sexual reproduction in the bacteria. Sexual reproduction, in the true sense, we can say that in the sexual reproduction the male gamete and the female gamete is formed. Then the fertilization process, that is the fusion of the male gamete and female gamete, takes place, and ultimately the formation of the diploid cell takes place. That is, we call it as a zygote. Okay, that is the process of the sexual reproduction. In sexual reproduction, what happens during the fusion of the male gamete and the female gamete, the genetic material, it get transferred from one cell to the another cell. Though in the bacteria the formation not reported, but the transfer of genetic material from one bacterial cell to the another bacterial cell is reported. Okay, and the sexuality in the bacteria, it was reported for the first time by the Tatum and the Laderberg in the 1947. Okay, they were working on the bacteria Escherichia coli. Okay, they were working on the bacteria Escherichia coli, and while working on this E. coli bacteria, they reported the sexuality, that is the transfer of genetic material from one bacterial cell to another bacterial cell takes place. Okay, here they reported that here it does not involve the fusion of the gametes. Okay, though the fusion of gametes never takes place in the bacteria, but the interchange of the genetic material, or we can say the transfer of genetic material from the one bacterial cell to the another bacterial cell takes place. Okay, and that's why the sexual, instead of referring it as a sexual reproduction, it is referred it as a genetic recombination. Okay, it is more appropriate to use the word genetic recombination instead of sexual reproduction in the bacteria. That's why I have mentioned in this slide the term genetic recombination. Okay, because it does not involve the fusion of gamete, but the interchange of the genetic material is reported. That's why it is more appropriate to refer it as a genetic recombination than the sexual reproduction. The genetic recombination, or the interchange of genetic material in the bacteria, it takes place by the three methods. It takes place by the three methods. The first one is the conjugation, second one is the transformation, and the third one, it is the transduction. Okay, three methods of the genetic recombination. The first one conjugation, second one transformation, and the third one is the transduction. Okay, of these three methods, in the conjugation, the two bacterial cells comes in close contact with each other. Okay, in the conjugation method, the two bacterial cell come in close contact with each other. But in the remaining two methods, the transduction and the transformation, the two bacterial cells never come in close contact with each other. Okay, that is the difference between these three methods. Okay, don't forget that in the conjugation, the two bacterial cell come in close contact with each other, but in the remaining two methods, transduction and the transformation, the two bacterial cells never come in contact with each other. Okay, we will discuss all these three methods one by one. Okay, here in this slide you can see the first method of a genetic recombination, and that is the conjugation. Okay, the conjugation, genetic recombination in the bacteria, it takes place by this method in the several bacteria. Okay, the genetic recombination or the interchange of genetic material by the conjugation method, it takes place in the several bacteria, but this method, it is best studied in the E. coli bacteria. Okay, it is best studied in the E. coli bacteria. Okay, here the mechanism of DNA exchange, or we can say the mechanism of the DNA exchange, it is mediated by the plasmid. Okay, here the genetic recombination between the two bacterial cells, it is mediated by the plasmid. Okay, we have discussed in detail regarding the plasmid. All you know that the plasmids, these are nothing but the extra chromosomal hereditary determinants. These are not playing any important role in the growth and viability of the bacterial cell, and that's why the genes which are present in the these plasmids, that is, these are said to be the these are the non-essential genes. Okay, these are not having the genes which are playing an important role in the growth and viability of the bacteria, and that's why the genes which are present in the plasmid, these are the non-essential genes. Okay, then there are about 9 different types of the plasmids up till now are reported. Of this one, the most studied plasmid, it is the F factor. Okay, here you can see the bacterial chromosome is present, and here this is the extra chromosomal hereditary determinant, and it is the F factor. F factor means the genes, that is, these are determining the maleness in the bacteria. F factor means fertility factor, which is determining the maleness in the bacteria. Okay, here there is some of the plasmids, some of the plasmids, these are self transmissible. That means they can transfer from one bacterial cell to the another bacterial cell on their own. Okay, some of the plasmids are self transmissible. That means they mediate their own transfer from one bacterial cell to the another bacterial cell. Okay, and also that is they can transfer the other plasmids. Okay, the bacterial cell which is having such self transmissible plasmid, these are said to be the donor. Okay, here you can see this is the bacterial cell which is having the F factor, which is self transmissible, and that's why this bacterial cell, it is referred as the donor. And the bacterial cell which is receiving this plasmid, this bacterial cell is referred as the recipient cell. Okay, it is called as the recipient cell. After receiving this plasmid, this recipient cell, it will get converted into the donor cell. That means this recipient cell get converted into the donor cell, here you can see, and ultimately this cell, it again come in contact with the another cell to which this plasmid, it is donated. Okay, it is the donor plasmid, means what, which is having the this transmissible plasmid, and the that donor cell and the recipient cell which is receiving that plasmid. Okay, at the end, due to the conjugation, that recipient cell, it is again get converted into the donor cell. Okay, some of the plasmids, that is, they lack the genes for the transfer. Okay, just I have told you that the some of the plasmids, these are self transmissible. That means these are having the genes which can mediate their own transfer. Okay, but some of the plasmids lack the genes for the transfer, and that's why such plasmids, these are transferred by the other plasmids which are carrying the genes necessary for the transfer. Okay, some of the plasmids lacks the genes for the transfer, and therefore their transfer, it is mediated by the other plasmids which are carrying the genes necessary for their transfer. Okay, in this diagram, in this photograph here, you can see the bacterial cell which is having the plasmid, which is having the F factor here. It is referred as the F positive cell. It is the donor bacterium. Okay, the bacterial cell which is having the F factor, which is having the F factor plasmid, it is referred here as the F positive cell. It is the donor bacteria, and analogous to the sexual reproduction in the higher organisms, here F positive cell, it is also referred as the male cell. Okay, the bacterial cell which is having the plasmid, the transmissible plasmid, it is referred as the F positive cell. It is called as the donor bacterium cell, and analogous to the sexual reproduction in the higher organism, this donor bacteria, it is referred as the male. Okay, while here this one, this bacterial cell, it is not having the F factor. Okay, it is not having the F factor, and that's why this bacterial cell, it is here referred as the F negative cell. It is receiving the plasmid from the F positive cell. That's why it is referred as the recipient bacterium, and F negative cell here, it is referred as the female bacterium. Okay, here F negative cell, it is referred as the female bacterium. Okay, here F positive, F factor is present, and F negative means the F factor is absent. Okay, F positive, it is the donor bacterium, it is the male bacterium, and the F negative cell, it is the recipient bacterium, and it is the female bacterium. The two most important thing in the conjugation are, the first one is the direct cell to cell contact is essential. Okay, here you can see the two bacterial cells, the F negative cell and the F positive cell, that is, these are coming in close contact with each other. That is the first most important for the transfer of genetic material, or for genetic recombination in conjugation. The two bacterial cell must come in close contact with each other. Okay, that is the first condition. And another important thing is that the conjugating cells must be of opposite strain. Okay, conjugating cells must be of opposite strain means what? Here you can see that the conjugation, it takes place in between the two opposite strain, that is the F positive cell and the F negative cells. Okay, these are the two most important things in the conjugation: cell to direct cell to cell contact, and the conjugating cells must be of the opposite cell. Okay, here you can see that the first step in the conjugation, the two bacterial cells of the opposite strain, F positive and F negative, they come in close contact with each other. Okay, the cell to cell contact, it is established by the formation of the sex pili. Okay, here you can see that is, these are connecting with each other with the help of the sex pili, and these sex pili, that is, these are forming a channel between the two cells. Okay, a channel is developed on the sex pili between the two cells. Okay, then here you can see in the third photograph here, that is the plasmid, it breaks down at a certain point. Okay, the plasmid, it breaks down at a certain point, and that certain point, it is referred as the ORI. Okay, ORI and capital T, it is referred as the ORI. That means that is the origin of the transfer. Okay, the plasmid DNA breaks at a certain point, and that point, it is called as the origin of the transfer. That is referred as the origin of the transfer. Then what happened? The 5 dash region, here you can see the 5 dash region of the double stranded DNA, it enters into the recipient bacterial cell. Okay, here the 5 dash region of the double stranded DNA, it enters into the recipient cell. Okay, and then in the recipient cell there is a immediate replication, immediate copying on the template strand in the donor cell in the 5 dash to 3 dash direction. Okay, there is a immediate copying of the template strand in the donor cell in the 5 dash to 3 dash direction takes place. And ultimately later on here you can see the it will restore its double stranded nature. Okay, in the donor cell that will again restore its double stranded nature, and it will assume the closed circular form. Okay, it will restore its double stranded nature, and later on it will resume its circular form. Okay, and thus here you can see the F negative cell, it get converted now into the F positive cell. Here you can see the F negative cell now becomes the F positive cell. Okay, that F positive cell then come again in contact with the F negative cell and will transfer it plasmid to the F negative cell. Okay, that is how exactly the conjugation takes place. Okay, here you can see during conjugation a cytoplasmic connection forms between the F negative and the F positive cell. Then here in this one you can see the F1 plasmid replicated through the rolling circle and transfer to the F negative cell. Then here the F negative bacterium, the single strand replicates, means the copying of the genetic material, it will takes place into the F negative bacteria. That's why it will retain its original double stranded nature, and later on producing a circular double stranded copy of the F plasmid. And lastly the F negative cell, it now becomes the F positive cell. That is how the genetic material, it is transferred from the one bacterium cell to the another bacterium cell via the plasmid or with the help of the plasmid, and this method it is called as the conjugation. Okay, here the conjugation between the F positive and the F negative cell takes place. Okay, now here it is the another method of the F positive, another method of the conjugation. Here you can see the F positive cell is present. Okay, in the last photograph we have seen that where the plasmid, that is it is separately present, that is it is not integrated with the bacterial chromosome. Here you can see that is the F1 positive cell. Okay, but sometime what happens, the F1 plasmid, it spontaneously integrates with the chromosome and thus converting this F positive cell into the HFR cell. Okay, sometimes the plasmid, it spontaneously get integrated with the bacterial chromosome. Here you can see the dark black colored, it is the bacterial chromosome, and this light colored, it is a part of the plasmid. Okay, and such integrations, the cell, the bacterial cell in which such integration takes place, these bacterial cells are referred as the HFR cells. The long form of HFR, here we can see that it is the high frequency recombinant cell. Okay, the HFR cell means what? Due to the integration of the plasmid into the bacterial cell, this bacterial cell, it is referred as the high frequency recombinant cell. Okay, then high frequency recombinant cell, it will come in close contact with the F negative cell with the help of the sex pili. Okay, the contact, it is established between the HFR cell and the F negative cell with the help of the sex pili, and this sex pili, they will form a small channel. Sometimes this small channel, it is also referred as the genetic tube, because the genetic material, it is transferred through this channel. Okay, the sex pili, that is it will develop into the small channel which is referred as the genetic tube. Okay, that means the conjugation of HFR cell and the F negative cell begins. Okay, here you can see the parental strand, it will get a breakdown, and along with the some amount of the plasmid, it will also get breakdown, and through the genetic tube, this genetic material, it will get transferred into the F negative cell. Okay, here you can see it will get transferred into the F negative cell. The HFR cell chromosome, here HFR cell chromosome replicates, and the parental strand of the chromosome, it is transferred to the recipient cell. Okay, and the transfer of small f factor also takes place along with the parental strand. Okay, now the piece of that chromosome, the piece of that donor chromosome, here you can see, here it is present, the piece of the donor chromosome. Okay, that piece of the donor chromosome, then it will get integrate with the bacterial chromosome of the F negative cell, and thus that F negative cell, it will get converted into the high frequency recombinant cell. Okay, the recipient cell, it receives the part of the plasmid and the chromosome, and then the transferred chromosome fragment exchanges genes with the chromosome of F negative cells by the recombination, and it confers the new property. That means that F negative cells, it will get converted into the high frequency recombinant cell. Okay, that is how the that is how the conjugation takes place in the bacteria. Okay, here the two most important thing in the conjugation are that the when we compare the conjugation with the other two methods like the transformation and the transduction, the relatively higher amount of DNA, it is transferred in the conjugation. Okay, when we compare the conjugation method with the transformation and the transduction, in the conjugation, relatively higher amount of DNA, relatively higher amount of genetic material, it is transferred. Okay, and that's why the conjugation, it is very important in increasing the genetic diversity in the bacterial cells. Okay, I am repeating this one. The relatively higher amount of DNA, it is transferred in conjugation than the transformation and transduction, and that's why the conjugation method, it is important in increasing the genetic diversity in the bacterial cells. Okay, that is all about the conjugation. Now, the second method of the genetic recombination in the bacteria, it is the transformation. Okay, transformation, it is a very unusual method because in this one, the two bacterial cell never come in contact with each other. Okay, just like in the conjugation. Okay, the two bacteria, it is not necessary that the two bacterial cell come in close contact with each other. That's why here we are saying that the transformation, it is a unusual method. Okay, here the DNA which are carrying the genetic information, these are transferred to the donor cells by the recipient, transferred from the donor cell to the recipient cell through the liquid medium. Okay, here you can see through the liquid medium, here these are the DNA fragments which are present in the liquid medium, and these are then transferred into recipient cell. Okay, that means the conjugation, it is the method in which the genetic recombination is mediated by the plasmid, but the transformation, it is the method of genetic recombination in which the transfer of genetic material, it is mediated through the liquid medium in which the bacterial cells are growing. Okay, this method, it was accidentally discovered, it was accidentally discovered by the Frederick Giffith. Okay, it was accidentally discovered by the Frederick Giffith in the 1921 while he was working on the bacteria Diplococcus pneumoniae in the mice. Okay, while working on the Diplococcus pneumoniae, Frederick Giffith in the 1921 accidentally discovered this method of genetic recombination in the bacteria. Okay, here the part of DNA, part of DNA having the quality of transformation. Okay, here you can see this is the DNA fragment, these are DNA fragments. Okay, it is not necessary that all these fragment, that is they get entry inside the bacterial cell. Okay, they get transferred into the bacterial cell. That is only few of them are having the quality of transformation. Okay, the part of DNA which is having the quality of transformation is referred as the transfer factor. Okay, it is referred as the transfer factor. And the bacterial cells which are having the ability to take the DNA, to take the genetic material from the outer environment, from the external environment, these bacterial cells are the competent bacterial cells. Okay, the bacterial cells having the ability to take the DNA from the external environment, these are said to be the competent bacterial cell. Okay, some of the bacterial cells, these are naturally competent. Okay, these are naturally competent, that means that is they does not require the any special treatment for the transformation. These are naturally competent, and that's why they does not require the special treatment to receive the genetic material from the external environment. Okay, during this process the rigid cell wall, here the cell wall of the bacterial cell, it is very rigid, and during this process the rigid cell wall can transport the relatively larger DNA molecules. That's why we are saying the transformation, it is very unusual method. Okay, as the rigid wall of the bacterial cell, it can transport the relatively larger DNA macromolecules. It is said to be the unusual method. Okay, it is not necessary that all the bacterial cells are naturally competent. Okay, it is not necessary that all the bacterial cells are naturally competent. Okay, then the bacterial cell, these recipient cells may take maximum of about 10 fragments of the foreign DNA. Okay, the recipient cell may take maximum of about 10 fragments of the foreign DNA. Okay, here the you can see the first step in this process is the entry of this exogenous DNA inside the bacterial cell. Okay, that is these DNA fragments will reach the entry site. Okay, they will reach the entry site, and then due to the nuclease enzyme, these are double stranded DNA, it will get breakdown, it will get converted into the single stranded DNA. Okay, when the exogenous DNA reaches the entry site, then this DNA fragment, double stranded DNA fragment, it will cut away, it will cut down by the nuclease enzyme into the single stranded DNA. Okay, and thus only one strand of the DNA molecule, it enters through the minute pores or the sites on the cell wall. Okay, only the single strand of the DNA, it will enter inside the bacterial cell through the minute pore or the sites which are formed on the cell wall. Okay, once they get entry inside the bacterial cell, the portion, it will combine, the exogenous DNA, it will combine with the bacterial chromosome, and that is called as the stable transformation. Okay, that exogenous DNA which is entered into the bacterial cell, it will get incorporated into the bacterial chromosome, and it will called as the stable transformation. But sometime what happens, once it will get entry inside the bacterial cell, if it fails to integrate with the bacterial chromosome, the degradation of these single stranded DNA takes place due to the enzymes which are present in the bacterial cell, and that's why it will not get integrated with the bacterial chromosome, and that is the unsuccessful transformation. Okay, that is we called it as a unsuccessful transformation. Okay, the generally the this transformation, it takes place under the special conditions. Okay, and these special conditions are referred as the state of competence. Okay, the transformation, it takes place under the special condition, and it is referred as the state of competence. And the state of competence of the bacterial cell, it may last due to the high temperature, ultraviolet radiations, and the use of the certain type of the chemical mutagens. Okay, the state of the competence of the bacterial cell, it may last due to the high temperature, ultraviolet radiations, and the chemical mutagens. Okay, while if we add the certain type of the compound, for example, if we add the inorganic phosphates to the nutrient medium, then the frequency of transformation will be increase. Okay, addition of inorganic phosphate to the nutrient medium will increase the frequency of transformation. Generally, the frequency of transformation, it is depending upon the temperature, then composition of nutrient medium, and the physiological state of the recipient cell. Okay, the frequency of transformation, it is depending upon the temperature, composition of nutrient medium, and the physiological state of the recipient cell. Okay, then the optimum temperature which is required for the transformation, it ranges from 29 degree to the 32-degree centigrade. Okay, the optimum temperature for transformation, it ranges between 29 to the 32-degree centigrade. Okay, then there is a difference between the conjugation and the transformation, is that in the conjugation, direct cell to cell contact is essential, and it is mediated by the plasmid. But in the transformation, direct cell to cell contact is not essential, and the genetic recombination, it is mediated through the nutrient medium. Okay, then another difference is that the transformation, it takes place between the members of same species of the bacteria. Okay, transformation, it takes place between the members of the same species of the bacteria, while the conjugation, it takes place between the two different genera. Okay, conjugation, it takes place between the two different genera. Okay, then the conjugations, that is, these are the temporary genetic recombination. Okay, these are temporary. We have just discussed, that is, the recipient cell ultimately get converted into the donor cell after the conjugation process get accomplished. Okay, but the genetic recombinations which takes place in the transformation, these are permanent. Okay, in the conjugation, the genetic recombinations are temporary, while in the transformation, the genetic recombinations are permanent. Okay, and now the third one, the third and the last method of genetic recombination, it is the transduction. Here, the transfer of genetic material from one bacterial cell to the another bacterial cell, it is mediated through the bacteriophages. Okay, it is mediated by the bacteriophages. These are the viruses which are attacking the bacterial cell. Okay, here you can see the bacteriophage, that is, it get absorbed on the bacterial cell. We have discussed the multiplication in the viruses. Okay, we have discussed the multiplication in viruses, and multiplication in the viruses, it takes place by the two method. The first one is the lysogenic lytic method, and another one, it is the lysogenic method. Here you can see it is the lytic method of multiplication of the viruses. Here you can see the bacteriophages, these are adsorbed on the bacterial cell. Later on, the genetic material of the bacteriophages, that is, it will get entered here into the bacterial cell. Then here you can see, then here it is the phaser DNA, that means the DNA of the virus, DNA of the bacteriophage. It will take control over the protein synthesizing machinery of the bacterial cell, and it will then direct it to develop the large number of the virus particles. Here you can see the while taking the here the while the multiplication of the virus, it takes place in the bacterial cell. What happens, the small piece of the bacterial chromosome, Here you can see it is a dark black colored, it is a bacterial chromosome, and here it is pink colored, it is the phage DNA. While their multiplication, what happens? A small piece of the bacterial chromosome, it will get incorporated in the attacking phase particles. Okay, here in this one is photograph, you can see the black colored pieces of the bacterial chromosome. These are incorporated into the phase particles. Okay, and then after which, after the multiplication of the viruses, what happens? The bursting or the rupturing of the bacterial cell takes place. Ultimately, all these phase particles are liberated in the outer environment. Okay, and then what happens? These bacteriophages, which are having the small piece of the bacterial chromosome, they will infect another bacteria. Okay, they will infect another bacteria, and ultimately that small piece of the bacterial chromosome, it will be released into the new bacterial cell. That is how the genetic material of one bacterial cell, it is transferred to the another bacterial cell via the bacteriophages. Okay, and that method is called as the transduction. Okay, that is all about the three methods of the genetic recombinations in the bacteria. Okay, three methods of the genetic recombination in bacteria. The first one is the conjugation. Direct cell to cell contact is required. Then the remaining two, in the transformation and the transduction, direct cell to cell contact is not required. Okay, in conjugation, the transfer of genetic material, it is mediated via plasmid. In the transformation, the genetic recombination, it is mediated via nutrient medium, in which the two bacteria are present. And the third one, the transduction, the genetic transfer from one bacterial cell to another bacterial cell, it is mediated by the uh, bacteriophages. Okay, the transduction, it was firstly reported by the Zinder and the Laderberg in the 1952. Okay, the transduction, it was reported by the Zinder and the Laderberg in the 1952, while they were working on the bacteria Salmonella typhimurium. Okay, they were working on the Salmonella typhimurium. Okay, that is all about